Hello, thank you for joining me. My name is Andy Kim, and I support tech transfers as part of the MSAT team here at Samsung Biologics. For today's presentation, I want to talk about some of the key considerations for enabling successful tech transfer. Manufacturing at large scale always comes with some level of risk. But the key question is, what steps can we take to reduce that risk? Here at SBL, the MSAT team works proactively with clients to identify and reduce risks. I'm sure many of you are familiar with this slide showing SBL's capabilities. Just if you haven't seen it, in brief, SBL has 3 plants here in Korea. Plant 1, the oldest plant, has 6 5K bioreactors. Plant 2 has 10 15K bioreactors. And Plant 3 has 12 15K bioreactors. We also have 1,000 liter bioreactors on site, both in single-use and stainless steel format. SBL is not just about manufacturing capability. We also have fully outfitted MSAT labs that can meet all your development and manufacturing needs. Now, the list shown here on this slide is by no means comprehensive nor fully detailed, but it's meant to give you a flavor of what we're capable of. So in the upstream space, we can perform microscale experiments using instruments like an Amber 250. Our typical scaled-down cell culture batches are performed using 10-liter bioreactors. And we can culture, harvest using 200-liter bioreactors. In the downstream area, we purify these materials using both active onts and active pilots for active scales. We use columns ranging in size up to 10 centimeters in diameter. And we also have UFDF equipment at both bench scale and pilot scale. We can also do depth filtration at pilot scale as well. And when it comes to our analytical lab, it's fully equipped with all of your standard equipment. We have HPLCs, UPLCs, PCR, CE instruments. We can perform assays ranging from protein titer, ELISA, CSDS, reduced, non-reduced, etc. The MSAT labs here analyze all the samples that are generated by both upstream and downstream to support our study. Now, there are many components to successful tech transfer. One that's often overlooked are the small-scale activities that take place. When a new project comes to SBL, we recommend typically perform verification runs at small scale. Runs allow us to de-risk process gaps before large-scale manufacturing, as well as to identify hidden risks. Once we're ready for manufacturing, MSAT would perform satellite runs at small scale concurrent with manufacturing. These are done using the same raw materials, the same buffers, intermediates. These runs are used not just to confirm what's seen at manufacturing scale, but they can also serve as a troubleshooting tool for investigations. Finally, throughout the life cycle of a project, we can also perform small-scale experiments for any reason needed. We perform experiments to support deviation investigations, as well as to evaluate post-manufacturing modifications to the process or mitigation actions. So oftentimes, when it comes to verification runs, a client will ask, why do we need to do these? And there's a couple of reasons I'd like to highlight. First, the verification run allows us to demonstrate that we have sufficient understanding of the client process when we can show that we can successfully execute the runs, even though they're at small scale. Looking at the bigger picture, verification runs are also a useful tool, offer their greatest value in minimizing risk or reducing risk prior to large-scale manufacturing. That's the key value. So looking at a case study for one of our clients, virtual biotech, you can see how verification runs are useful to achieve several aims: reducing risk due to known gaps, evaluating process changes, and identifying potential issues. Talking about risk mitigation and risk identification. In the first example, we were informed by the client that they have a limited number of old working cell bead vials. For our upcoming manufacturing campaign, we would need to use new WCB vials. To mitigate risk, the MSAT team performed several cell culture batches side by side using both the old and new WCB vials. These harvests were purified through Protein A, and the product quality was compared between the old and the new WCB. What we found was that the product quality measured by HMW, HCP, and DNA were found to be comparable. And the only real difference was in terms of performance, where interestingly, the new WCB vials offered improved protein A yield. Ultimately, we were able to deem that the new working cell bean vials were fit for use in our manufacturing with no risk. In the second example, we identified an unknown risk during the execution of the UFDF step of our downstream verification run. So during the UFDF step, when we tried to achieve the client-specified UF2 target concentration, which is much greater than 200 grams per liter, we encountered viscosity issues that were so severe we were unable to forward process. We also lost a lot of product. As a result of this, we recommended for manufacturing to target no higher than 200 grams per liter during the UF2 part of done without issue at manufacturing. And as a follow-up, we also looked at development reports, and we were able to realize that historically the client had never achieved greater than 200 grams per liter at their sending site or in development studies, despite their range established. So this is one good example of how we identified a risk before manufacturing and were able to prevent that risk from causing an issue at large scale. So small scale runs can be used to de-risk gaps or to evaluate potential modifications. In this case, for the same client, we're able to identify a risk with our pool vessel assignments where the client process requires a 1-to-1 dilution with Wi-Fi prior to that filtration. By following this procedure, we would have an intermediate pool volume greater than 10,000 liters right after the protein A step. But because our 15 kV vessels are located at the end of the hull, farthest away from the protein A skid, we would have to use a lot of hosing and a very complicated flow path. So to reduce the complexity that you see here in red and to increase process robustness, MSAT recommended a modified procedure, keeping in mind being aware of the process controls. Well, here being a conductivity check prior to the second column after dilution, where we check conductivity with spirits within 4 to 5.5 millisiemens per centimeter. What did we recommend? Our recommendation results in a simpler flow path, which you see in red here. And what we recommended was a 50% dilution with Wi-Fi, not 1-to-1. To evaluate the risk of this proposal, we evaluated it at small scale during our verification run, and we found that there was no impact to product quality or process performance. We also double-checked, are we capable of meeting our key control for connectivity? And although the resulting connectivity was higher as a result of this modified procedure, it was well within the established range, and the overall impact to the process capability was very minimal. So this was good news all around, is what we executed in manufacturing. As a final check mark on our checklist, we also performed a filter sizing experiment using the more concentrated, less diluted material. And as a result of that, we recommended a 30% decrease in dead filter capacity. So earlier, I mentioned that it is our typical practice to perform satellite runs concurrently with manufacturing. In this example here, for an engineering run, we found great value in the satellite runs. First, what happened in manufacturing? During the harvest operation for this batch, we first encountered significant filter clogging of the styro filters in harvest right after centrifugation. This was followed by even more severe clogging of the protein A guard filters. It was so bad that we had multiple filter changeouts. and ultimately lost more than 20% product. We also observed a 25% product loss in a subsequent chromatography step, and which at the time we did not realize were connected, but through our investigation, we were able to determine that they all had the same root cause. In that same group of issues, we also observed elevated levels of HAF antibody, noted by HL here on the slide, and high levels of HMW. These product-related impurities were seen throughout the process. So, looking at the satellite runs that were performed in conjunction with this manufacturing batch, not only did the satellite runs confirm the aberrant results we saw at manufacturing scale, but they gave us an early look at the product quality. So first, here on the bottom half of the slide, I have a chromatogram for one of the chromatography steps, comparing the verification run that we performed prior to manufacturing and the satellite run performed concurrently using the same materials from manufacturing. Same buffers, same process intermediates. The satellite run in pink, you can see significant change in peak shape for the flow-through peak, and you can also see a much larger strip peak. The manufacturing data is not shown here, but is identical to the satellite run data. When we looked at product quality, shown on the right-hand side, we have HAF antibody results on the top right half, HMW results on the bottom right half. What we can see for the satellite run results, colored in blue, they significantly differed from the quality results from the verification run, where we saw about 30% HAF antibody throughout the steps versus 3% for the verification run. For HMW, it reached levels of about 20% compared to less than 1% for the verification run. Benefit here was because we performed the satellite runs concurrently, as soon as the samples were ready, they were submitted to the analytical team and the results came out right away. Once the engineering run results were available, we submitted those to the analytical team, and those results came out to be almost the same and confirmed our earlier suspicions. There is one difference to note in the quality data. For the proteinase step, there is a difference between the satellite run and engineering run. The reason for that is due to a scaling factor, which prevents replicating the same behavior at small scale. The key message for the first step is that there is a negative product quality impact, even though the level of impact is different in the satellite run and engineering run. So in sum, the satellite runs not only confirmed what we saw in manufacturing scale, but they helped us to determine that there are no operational execution errors. The raw materials were good on further investigation. Buffers were prepared correctly. Equipment was in good operating order. Operators followed procedures, and no hold times were exceeded. So looking at these data and all the information that we have, we could not identify a root cause in the downstream process. This pointed to upstream events. By following up in our investigation with more small-scale experiments, we were able to determine an issue with antibody reduction due to unanticipated enzymatic activity. So this is a process issue that was not identified by clients, nor was it discovered during the development work, because it was manufactured at a much smaller scale. As further follow-up and looking ahead to what we could do to resolve this, we also performed experiments to evaluate different mitigation actions. We looked at potentially processing downstream at low temperature and also controlling dissolved oxygen levels to prevent enzymatic activity. But those experiments and those results are a story for another day. In conclusion, hopefully I've demonstrated to you the value in performing verification runs and satellite runs and how these small-scale activities can help reduce risk for large-scale manufacturing. In our case study, the verification runs were useful in identifying viscosity issues with an overly high UF2 target concentration. And we also demonstrated the comparability between old and new WCB vial sets. We also evaluated a modified dilution strategy to simplify vessel assignments and our overall procedures. The satellite runs were useful for troubleshooting and getting an early look at product quality. And that early look is important because it enabled us to start our investigation right away to look in the right places and find a solution more quickly. So one message I have for you, though, is these runs may be valuable, small-scale work is valuable, but to really realize the true potential, the benefits these runs may offer, requires a strong technical foundation. So my recommendation to you is when you select a CMO, find someone who offers to you the right balance between technical capability and technical expertise. So with that, I will wrap up with some concluding slides. This is a reference for you to check out the various service offerings here at SBL. And our last slide is an invitation to you to visit our virtual exhibition. Please take a look. Thank you and have a nice day.